Hi everyone, welcome to my YouTube channel. Today our topic is blood and plasma proteins. And before that, like, share, subscribe, comment down below, and I'm going to go right into it. Let's get into blood and plasma proteins. While we're going through this chapter, I'm going to take you through the set of questions that has been asked in the previous years. We will keep more focus on that particular topic. Okay, so what do we know? Blood and plasma proteins. But before we jump in, let's just take a look at the concepts that we will learn. We are going to learn about the composition of blood. Then we're going to learn the plasma normal volumes of blood, the plasma proteins and their functions, plasmapheresis, functions of blood, and blood composition and principles to identify blood. Now, why have I written this in separate topics is because they have been asked previously. Okay, now blood and its composition. As we know that blood is a fluid connective tissue. Hey, we know all this part, but now what is the key term over here? That it comprises 8% of the total body fluid. So it is a connective tissue that comprises 8% of the total body weight. Now, composition of blood, that is the plasma component and the formed elements component. Now, the plasma component contains like 91 to 92% of the total blood volume. Like as you can see here, 90 to 92% of water. It also contains albumin, globulin, and fibrinogen. Albumin comprises about 55% as a whole of the total blood. We'll learn about that later, but albumin is like the star protein. Now, composition of blood. Plasma comprises 55% of the total blood volume. And formed elements, which contains RBCs, WBCs, and the platelets, they comprise the formed element part of the blood, and they are about 45% in total. Now, if we take blood and we centrifuge it, there are going to be formed three layers that are plasma, buffy coat, and erythrocytes. Now, our main significant layer over here is the buffy coat layer, which comprises less than 1%. It is like a white thin layer that is formed by the above plasma layer and the below RBC layer. So this buffy coat that you can see over here, it mainly consists of platelets and the leukocytes. By the way, this is like a beautiful cute diagram that I have taken the reference from the internet. So, okay, the significance of the buffy layer. It helps in the identification of virus, fungi, bacteria. Now, the presence of primitive blood cells, they indicate that there are certain malignant and pre-malignant conditions that are present in the blood. Basically, it says that your blood is abnormal. That's all you've got to learn about the buffy layer. The normal values of the blood. This has been asked multiple times in your previous years, and not just that, it plays a very important role in your practical viva. Now, we have adult males, we have 4.5 to 6 million per cubic millimeter of blood. All right, so here these are certain set of values that you've got to keep in mind, and you've got to register it in your mind throughout the first year, throughout your whole life, that you're going to be pursuing MBBS in. If not, well, you will. So in adult males it is 4.5 to 6, and in adult females it is 4 to 5.5. All right. Now, in adult females, it is slightly less than the males. It is mainly contributed due to the presence of the estrogen hormones. Now, if you see in newborns and children, the newborns have like 6 to 8 million per cubic millimeter of blood, and children have 3.5 million per cubic millimeter of blood. So you can see that with advancing age, that is newborns from 6 to 8, eventually as a person hits adulthood, the blood value keeps decreasing to around as over an average of 5 million per cubic millimeter of blood. Now, why is that? The newborns have a very high amount of normal blood value, that is 6 to 8 million per cubic millimeter of blood. That's because in newborns, they tend to live in a low oxygen, that is the hypoxic environment, that is the placenta. Now, in order to compensate the hypoxic region, that is less amount of oxygen that is present in the placenta. So there is more RBC production. That means more amount of erythropoiesis takes place. Erythropoiesis is the formation of RBC, which is in order to compensate the hypoxic condition of the placenta. Another factor could be the fetal hemoglobin is HbF. Now, HbF has a pretty good affinity for oxygen. Alright, now our topic is plasma proteins and their functions. This is a super important topic and has been asked as a 10 marker. Now, overview of the plasma proteins. Now, albumin, fibrinogen, globulins, and the other regulatory proteins are the different types of plasma proteins available. Alright, so the albumin comprises 55%, fibrinogen comprises of 7%, and globulins comprise of the 38%. Now, albumin is like a very important and the abundant plasma protein that is available and has various functions such as maintaining the plasma oncotic pressure or the colloidal pressure. So, the osmotic pressure and transport of various substances. Fibrinogen is mainly related to the process of clotting blood. Globulins is mainly related to the immunity related or the defense function of the body. Albumin is about 55% of the total, fibrinogen is 7%, and globulins is the 38%. The other regulatory proteins are like the other enzymes, hormones, and other complement proteins that are present in the body which have various functions. So you can just represent it through this diagram. It is always lovely if physio you represent through flowcharts, points, graphs, and table format. Alright, now albumin, which is the most abundant plasma protein, it is mainly 3.5 to 5 gram per deciliter, exclusively synthesized in the hepatocytes of the liver. Alright, now the colloidal osmotic pressure. This was asked previously and it is a very important PYQ. Now, albumin is the primary determinant of the plasma colloidal osmotic pressure. Before I explain what that means, I want you to look at this point. It prevents edema and fluid leakage, these 2 points. Now, let's say that this is your capillary, and usually the capillary or whatever, it has the albumin. Albumin is like a pulling protein that tends to pull water from the interstitial space into the capillaries. As in, if this space that with red dots that I am marking is the interstitial space, alright, albumin tends to pull fluid from this interstitial space. Alright, in certain conditions such as liver cirrhosis. Why? Because if liver is the primary area where albumin is synthesized and liver is damaged, let's say there's low albumin. Now, because of the absence or hypoalbuminemia condition, there is nothing that will pull the fluid from the interstitial space. Therefore, it's going to cause the accumulation of fluid in the interstitial space. Eventually, this accumulation of fluid in the interstitial space is going to lead to a condition called as edema. Edema is nothing but a swelling like appearance that you see in the body because of the presence of fluid in the interstitial space. But now you know the main factor or the reason for such type of condition is because of albumin. The colloidal osmotic pressure is the pressure required by the plasma protein to pull fluid in back into the blood capillary. So it is mainly the function of the plasma protein albumin. It has another function called the transport function, where it helps in the transport of various hormones, enzymes, pharmaceutical drugs, thyroid hormones, fatty acids, etc. So it basically is a transporter. The clinical significance, which we just talked, is hypoalbuminemia leads to a condition of edema, especially when an organ such as a liver is destroyed. So it maintains colloidal osmotic pressure, transport like excuse me, transport ligands, and prevents edema. Alright, now globulins, diverse functions proteins. Now, globulins are like the immunities of the body. Their average number is 1.8 to 3 gram per deciliter, and it is also synthesized by the liver. Alright, so it is 1.8 to 3 gram per deciliter. This is the normal range of the globulins that is present in the blood, and they are divided into alpha, beta, and the gamma proteins. Okay, so now there are different types of alpha globulins and beta globulins. They are alpha anti-1 trypsin, alpha 1 glycoprotein, and alpha 2 macroglobulin. Similarly, in beta, there is complement proteins and the transferrin. We'll finish beta globulins. Transferrin. transport ferrin iron, so responsible for the transport of iron in the blood, delivering it to the cells wherever it's required. Complement proteins are like C3a, C5ab, etc. that are present. You'll learn that in the immunity chapter. They're mainly helping in like defense like functions. Now alpha globulins, alpha 1 antitrypsin, alpha 2 macroglobulin, alpha 1 acid glycoprotein. There are many more alpha, but then yeah. Hey, also, I remember the question I had in biochemistry Viva was what are the different types of alpha and beta globulins? Well, now I'm pretty sure all of these are of very huge use. Now, alpha 1 antitrypsin, it is mainly used in the treatment of lung-based disorders or diseases like emphysema, etc. Or like it is mainly protecting the tissues from the enzymes like elastase, especially in the lungs. Alright, the acid 1 glycoprotein binds to the transporter basic drugs and it is an acute phase reactant, basically again infection based. Macroglobulin, another large protease inhibitor. This is an extra point, but then no problem if you knew it. It's just like inhibits a type of enzyme that is the protease that is helping mainly in the transport. Alright. So gamma globulin, they comprise of IgA, M, D, and E. They are produced by the B lymphocytes and the plasma cells. And since it is produced by the B lymphocytes, it is related to the humoral immunity. Alright, now fibrinogen, unlike albumin in globulin, is mainly going to be related to the blood clotting function. Alright, where it is 0.2 to 0.45 and it is mainly synthesized by the liver. Alright, now you can see I'm going to explain this entire thing just through the help of the diagram. Now fibrinogen, which is synthesized in the liver, in the presence of the enzyme thrombin, gets converted to as the fibrin. This fibrin results in a mesh. Okay, now because of this fibrin, there is going to be like a mesh like formation on the area where there is clot. Now because this clot is formed on the area where there is a wound, so this clot prevents the leakage of the RBCs and the platelets, and therefore your wound is sealed and it's clot, where fibrinogen gets converted into fibrin due to thrombin and results in the clot formation. Alright, so that is what this entire thing, synthesis and structure, role in coagulation, and finally the formation of clot. Alright. Now the normal plasma proteins and the AG ratio. I don't have to tell you in detail about the plasma proteins because you already know their normal values by now. But the total plasma protein, this one, is typically 6 to 8 gram per deciliter. So if there is less than 6 gram per deciliter in the blood, the total blood concentration, it is hypoproteinemia, total protein. So hypoproteinemia. But then if there is more than 8, it is hyperproteinemia. Alright, so now we'll be talking about the AG ratio and its importance. It has been asked again in the PYQ. So the normal range of the AG ratio is 1.2 is to 1.5. Some places you will also find 2.2, but it is absolutely normal even if it is 2.2. Now the low AG ratio, usually A divided by G. Okay, in this case, A divided by G, let's say it is 1.2. Clearly, in this case, albumin is greater in concentration than the globulin. In certain conditions where there is low AG ratio, as you can see over here, it is because albumin is lesser in concentration than globulin. So AG ratio, let's say it is around 0.2. Now, if albumin is less than globulin, there could be two main conditions. Liver cirrhosis. Because liver synthesizes albumin, something happened to the liver, albumin is low. Second, nephrotic syndrome is a problem with the basement membrane where the albumin, instead of staying in the plasma, will be excreted out in the urine. Now, because this albumin is getting excreted out into the urine will have increased amount of albumin, but the plasma will have decreased amount. I mean to say, let's say this is the body of the human being, alright? Don't mind this diagram, but yeah, now you know that this is a human being. This is the urine. So let's say there is problem to the basement membrane that is related to the nephrotic syndrome. So what's going to happen? There is going to be albumin. Albumin is going to escape out. Why? Because the structure of albumin is very tiny already. So it is going to escape out into the urine. Now there'll be more albumin from the body. It has escaped into the urine. So what's going to happen? Albumin concentration in the body is going to eventually decrease and it is going to increase in the urine. So but right now, since we're talking about plasma and plasma is present mainly in the body, so we are going to say that since plasma is mainly present in the body, it leads to hypoalbuminemia or the low A concentration, that is low Ag ratio. Now also a high Ag ratio is indicated where the globulin production is less, and it could be mainly due to certain immunodeficiencies or the genetic disorders. All right. So what do we know next? Now the clinical significance of the plasma proteins. That's what I said. Hypoproteinemia and hyperproteinemia. If it is less than 6 and this is more than 8. So hypoproteinemia is mainly because there's less amount of proteins. That means less amount of proteins in the plasma, more amount of proteins in the urine. So it is mainly due to liver cirrhosis, malnutrition, or excessive loss, maybe due to nephrotic syndrome. That's what we've been discussing previously. Now, hyperproteinemia, it is mainly caused due to increased globulin production. Now, the serum protein electrophoresis is something extra simple, where I will take an electrophoric membrane and I will separate the proteins based upon their size and density. Now whatever goes first becomes the alpha, whatever is remaining beta, then gamma. So basically according to that it is divided into albumin, alpha 1, alpha 2, beta, gamma globulins based on their electrical charge and size. Alright. Now, in multiple myeloma, there is going to be a sudden spike in the gamma region. That is this region, gamma globulins, that is related to the infection region. There is going to be a spike in the gamma potential. In cases of chronic liver diseases, where the albumin will be less, there will be reduced albumin and elevated gamma globulins point towards the chronic liver diseases. Plasmapheresis. It is the process of removing plasma from the blood and returning the cellular components to the circulation. So basically, this is mainly performed, let's say, when I have harmful blood. Okay, harmful substances that are present in the plasma are going to be removed. So plasmapheresis is the process in which, okay, I remove the plasma from the blood, returning back the formed elements. Now what does that mean? Now you know that blood as a whole has two components under it. So one is the plasma component and the other one is the formed elements component, that is comprising of RBCs, WBCs and thrombocytes. Now I am going to remove this from the blood and return this formed elements back into circulation. Now when I do this thing, this process is known as plasmapheresis. Principle of measurement of blood volume. Now the amount of dye that is mixing with the blood once it has been injected, okay, it is equal to the volume of the blood. Understanding? I inject the dye, it mixes with the dye, whatever has mixed with the blood, it is equal to the volume of the blood, because if less amount of volume of blood is there, the dye that I am injecting, less amount of dye is going to mix with the blood. So that's what it means. A known quantity of indicator is injected into the circulation and allowed to mix evenly and freely with the blood. Its concentration after mixing can be used to calculate the volume in which it has been distributed. This over here is the blood. So the formula to calculate the blood volume is the amount of indicator injected divided by the concentration of the indicator after mixing. So this was not deemed of much importance, but you will have to know this concept. It is there, it exists. Okay, and this skeleton dude over here, it's just showing how deep fried our minds are. Not getting deep fried, and we'll meet you again in the next upcoming videos. And do like, share, subscribe again. Thank you. Bye bye. Have a nice day.